Hello, everyone. My name is Miles Roberts, and I'm a first-year grad student at Michigan State University, where my advisor is Dr. Emily Josephs. The study of adaptation is a primary focus of evolutionary biology. When evolutionary biologists study adaptation, there's often a focus on a single trait and how it contributes to fitness by itself. For example, one trait that plant biologists often study in its relation to fitness is flowering time. This trait is just defined as the number of days between when a seed germinates and when open flowers are produced on the resulting plant. It's an important component of the fitness of many flowering plant species. For instance, whether a plant has open flowers at a time when pollinators are around could be certainly influence whether or not a given plant can reproduce. However, another important life history trait that often evolves alongside and is correlated with flowering time is growth rate. You might imagine, for example, that plants with a shorter flowering time also experience selection to have high growth rates, such that during that shorter interval of time, the plants are actually able to achieve a size where they can produce flowers and offspring. That would make these two traits negatively correlated. However, what's interesting about these two traits in the model plant Arabidopsis thaliana, which is pictured here both times, is that the correlation between these traits varies across the range of A. thaliana, which is very peculiar. This was well demonstrated in a 2013 study by Debut et al., where the study authors looked at 112 different genotypes of Arabidopsis and looked to see how the correlation between growth rate and flowering time changed with latitude in this sample. And this is the result they found. So on the vertical axis is, again, the correlation between growth rate and flowering time. And the horizontal axis is latitude. You can see that as we go up in latitude, the correlation between these traits steadily declines. Now, we rarely understand the genetic basis for why trait correlations could differ like this between, in this case, low and high latitude Arabidopsis thaliana populations, or really just between populations in general. But if we did, then that could be really useful in, for example, identifying genetic variants that will break up undesirable trait correlations in plant breeding. So today I'm going to discuss an approach I've used involving genome-wide association models to chip away at what genetic differences between low and high latitude Arabidopsis populations could be causing this trait correlation cline. To start, we needed a larger sample than just 112 different genotypes, and I got that by combining data from two previous studies on growth rate and flowering time respectively. This map just shows the locations of where all those genotypes were isolated from. There are about 400 in total for this sample. And when I repeated the analysis on the previous slide using this new larger sample, we got a pretty different result. So on the vertical axis is still the flowering time growth rate correlation, and then a horizontal axis is still latitude. Instead of seeing a steady decline in the trait correlation, we see this sharp peak at around 48 degrees latitude or so. And I think there are a number of reasons why I'm getting a different answer here than that previous study. First off, the measure of growth rate I'm using is different than what the previous study used. And also my sample includes genotypes from a wider range of latitudes and longitudes. However, I still think that this trend is strong enough and interesting enough to warrant further investigation. So, but what can we actually do to detect whether there are differences in the genetic basis of these life history traits between low and high latitude Arabidopsis populations? There are a couple approaches I've used. I'm just going to talk about one today, though. And it starts by taking the individual Arabidopsis genotypes in my sample, which I'm representing with red dots here, and then ordering them by the latitude that they came from. And then once I have this ordered list of the genotypes, I can then break them up into overlapping subsets of data, progressively moving up the latitude gradient, which with each window containing 103 individuals. And then to each of these windows, we can apply a genome-wide association model that will look for genetic variants whose presence or absence in the genome is associated with altering our life history traits of interest, including the two that I just talked about, flowering time and growth rate, as well as this new trait, which is the first principal component from a PCA of these first two traits. You can think of this last trait as sort of a composite phenotype, combines aspects of both growth rate and flowering time. And having that in the model allows us to look for genetic variants that are associated with changing growth rate and flowering time simultaneously. When we apply all those models, these are the results we get. So the horizontal axis here is still latitude. The 0th interval is the interval of individuals at the lowest latitudes, and interval 19 contains the interval of individuals at the highest latitudes. The vertical axis just shows the number of genetic variants that were significantly associated with our life history traits of interest when this model was applied to each interval. I just have the data for flowering time plotted here. And you can see already that there's a lot of variation between the individual latitudes intervals and the number of genetic variants associated with flowering time. This one outlier interval up here has 2,000, while many of the others have several hundred. And the lowest latitude intervals have absolutely none. You see a pretty a similar story for growth rate, where these higher latitude intervals, again, have quite a few associations or genetic variants associated with growth rate, while the lower latitude intervals have much fewer. And then finally, for this last trait, which again combines aspects of both growth rate and flowering time, the mid to high latitude intervals now have lots of genetic variants associated with this trait, while still the lowest latitude intervals don't have many. So I think that this suggests that there could be some genetic differences in the basis of these traits between low and high latitude Arabidopsis populations that could be underlying the cline and trait correlations that we see. But what could be the actual mechanism going on here? What genetic changes aren't just altering our life history traits of interest, growth rate and flowering time? Which changes could actually be altering the correlations between those traits? One possibility involves a phenomenon called Pliotropy. This occurs when a single genetic locus influences more than one trait, and that often leads to a correlation between said traits. One way to alter a trait correlation that's caused by Pliotropy is through mutations in regulatory elements. So here I've added a circle with an X through it to represent a mutated regulatory element, and that mutation has affected trait 2, but not trait 1. You might expect to see a result like that because regulatory elements often have tissue-specific effects on gene expression. So if a gene is expressed in multiple tissues, mutation in one of its regulatory elements could alter how the gene is expressed in some tissues, but not others, thus potentially changing some of the traits contributed to by the gene, but not other traits. We think these types of genetic changes could be what's underlying the variation in trait correlation we see across latitude in A. thaliana. To test that, I'm going to take these results that I just show you. This is just the last graph that we saw. And then I'm going to look at what proportion of these significant variants fall in open chromatin regions. Open chromatin regions are just regions of the genome that are accessible to be bound by transcription factors, and thus they are tightly correlated with these sites of regulatory elements. And when we do that, this is what we get. So this vertical axis is now the proportion I just stated, the proportion of variants that fall in open chromatin. And the dotted line is the proportion of the entire genome that falls within open chromatin. And you can see, looking at these different intervals, some of the bars fall just above the dotted line, some fall just below, but overall there's not a whole lot of enrichment of these variants in open chromatin regions, which goes against our idea that mutations in regulatory elements could be the difference between low and high latitude A. thaliana populations causing the decline in trait correlations. You might be wondering about this outlier interval up here. That's just because this interval contained 2 significant variants, one of which fell in an open chromatin region. You can also see some drawbacks The data look kind of sparse because not every interval returned significant results in the GWAS model. And these are all just associations at the end of the day. So going forward, we want to apply alternative modeling approaches and benchmark those different approaches against simulated data sets to get a better idea of how they perform and hopefully shed more insight on what genetic changes actually alter trait correlations in nature. So thank you very much for your attention. I'd like to quickly acknowledge Joanne Schmidt, who gave me the idea to do this analysis, and of course, MSU and NSF.